hopefully.
Okay, so yeah, thanks for inviting me to this very different setting. So it was actually a very interesting exercise to try to relate my research to autocatalysis. And you will see that I try to make some links. I work with origin of life, and autocatalysis pops up all the time, but I had never really stopped to think about it that much. So before talking about autocatalysis itself, I thought it was perhaps good to discuss reaction kinetics in coacervate droplets in general, and possibly introduce the idea of coacervates to you. I'm not sure if you're already familiar, but coacervates are a type of chemical assembly that it appears throughout chemistry in a lot of different ways. One way is in origin of life research, and that was back when Oparin and Haldane, like Alex said, they were looking for an explanation to how prebiotic chemistry produced eventually primitive cells. And they proposed that you needed these colloidal structures to drive the increase in molecular complexity. Otherwise, that could not happen. And this is a very long paragraph from Oparin chapter on origin of life. But let's just look at the yellow parts. So he says, let us assume that in one of the corners of the earth, in the turbulent waves of the ocean, there were formed, either at the same time or one after the other, 2 bits of gel. And later he says, the one with a physical chemical organization which made it possible to carry out the process of assimilation of foreign substances from the environment more quickly also grew faster than its weaker, less well-organized comrade. And the more it grew, the larger its surface became, the wider became the difference in the rate of growth between these 2 gels. So what is interesting here is these 2 ideas. First, he refers to bits of gel, and I think at some point he started to use the term coacervate. But you have so it's just this idea of a compartment on a constant, a dense compartment. I would say. And when I read this, I see the idea of autocatalysis here, because he's essentially saying that you have this gel growing and becoming better at growing all the time, because it's just constantly taking up more material. And the reason why this is called the Coacervate theory is because at a similar time, there was a chemist in the Netherlands, Bungenberg de Jong, developing the concept of coacervation. So he discovered that there were these mixtures that instead of separating in a liquid phase and a solid phase, so that's very common, that you have a precipitation, for example, he discovered that there were mixtures that would separate in 2 liquid phases. So one phase mainly this being the solvent, and the second phase being a solute, but still retaining a lot of solvent, so it was still liquid. And he decided to call those coacervates. And he already saw that when this mixture is still organized as an emulsion, there was a lot of resemblance to the protoplasm, because the liquid phase is very dense and was taking up a lot of guest molecules. And so this is a really good chemical model for what Oparin and Haldane needed in their scenario. And a further indication that this is a good model comes from a bit more recent developments in chemistry. That is the discovery that this these type of structures are present in modern cells. So then you will normally see them called condensates instead of coacervates. But if you expand the definition of coacervates a little bit, you can say that they are the same. So there are these organelles inside the cell that they don't have a membrane, they fuse like liquid droplets. And since the discovery of these organelles, what's more interesting is that people found that several proteins, well, a majority of proteins in our proteome can adopt this droplet state. So this seems to be a property of biomolecules. There would be a reason to investigate these structures already. And we know that those structures are present, but we don't know exactly their function in all of the cases, and if they have any function. So with this, there's all these three reasons to study reactions in coacervates, because if you then have reactions incorporated in these compartments, it might have consequences for prebiotic chemistry, like Oparin expected. It has consequences for the phase behavior of these droplets, how they nucleate, how they grow, and how they are stable over time. And it has consequences for cellular biochemistry as well. Besides the concept of coacervate, another important concept is that of passive and active coacervates here. So that's I think in a way it will relate a lot to whether the droplets display autocatalysis or not. But so this is a concept for emulsions in general, and it applies just the same for coacervates. So passive coacervates are droplets composed of a substance B, for example, and they might have a reaction inside. But B and the solvent A are just expectators. They're not doing anything here. So these droplets always reach phase equilibrium, the concentrations of A and B. The reaction can reach equilibrium. These droplets behave like droplets that we know. They can fuse, they can wet surfaces. They undergo auto ripening, so over time you always form a single droplet or a macrophase. And because you can have this exchange between the droplet and the exterior of product and reactant or other molecules, you can also cross Marangoni flows. But this is pretty much it. When instead you have an active droplet, the requirement is that the reaction involves the component of the droplet. So here, this is an example by David Zwicker. You simply have a reaction that makes droplet material, which is B, and you even have a reaction that consumes that material. But so you have this constant formation of droplet material. So the droplet always needs to achieve phase equilibrium, and the reaction can be fueled as well. So you're always, you're in a way always out of equilibrium. In this case, it's because the droplet behavior is more interesting, let's say. So you can have altered Oswald ripening, either enhanced or suppressed. There's indications for both. The droplet will grow in a way controlled by the reaction. So that's not the case with the passive droplets. There is the prediction that these droplets can divide to create gradients that also propel the droplets. And finally, there's an understanding, I would say, that these droplets are, the droplets or the reaction here is an example of physical autocatalysis. And I think I'll discuss this a bit more. But the presumption is always that the droplets in both cases, in passive and active droplets, is that once there's a reaction incorporated, the droplets assist that reaction. So that's the presumption from Oparin's hypothesis, and often the case when people look at biological coacervates, so condensates, there's this assumption that droplets help reactions. So I think the first thing to look at is whether this is the case, whether coacervates can assist reactions. And that's also a requirement for physical autocatalysis, as I understand it. So first, let's look at reactions just happening in passive coacervates. So let's not worry about what happens to the droplet material. It's just there. And I will show you a bit of modeling work that I did during my PhD. So the way reactions can be different in coacervates is in general, it can be divided in maybe in two big categories, but here I'm showing four. But there's essentially a concentration effect and a transition state effect. So molecules can be concentrated inside the droplet. They can also be excluded, and this can lead to rate enhancement by itself, or you can also change the rate constant of a reaction inside the coacervate. So people generally assume that the environment of the droplet is very different than a solution because it's crowded, because it has a little bit less water. And this can be the case in particular for reactions that require some spatial conformation of the reactants. And consequences of this, of course, are that cascade reactions are very influenced by being in a compartment because they are essentially organized in the same space, so they will become more efficient. And another effect is that of product release. So if you think of the concentration effect, but if you consider both the reactant substrate and the product, if they distribute unevenly in droplet and the surroundings, there might be a cycling effect where you're essentially just removing product and driving the reaction to reach equilibrium faster or reach completion faster. But in all these cases, in all these examples, you can find examples in the literature, experimental examples, that will show either rate enhancement or rate decrease. So it's not enough to say that it is not really precise to say that coacervates help reactions because that has not been proven experimentally. So let's look a bit more in detail what can happen to a reaction taking place in this biphasic environment. So I'm showing here what I consider a very minimal model of a reaction, so, with a substrate and an enzyme forming product P. So this is relevant because of the biological relevance, of course, of coacervates. And then you have these rate constants here, the lowercase k's. There are diffusion or transport constants, the lowercase k with a t. And there is also the partition constant, the uppercase k, that are essentially the distribution of a molecule in the droplet relative to the outer solution. And the final parameters that describe the system are the volume of each phase. They are normally described by a single parameter, which is the volume fraction. So this is saying how much droplet material you have in an emulsion. So what we can do now is just look at what happens to a reaction as we change one of these parameters. So for example, it's very common that an enzyme will be highly concentrated in the droplet. And it's very common also that the substrate will be highly concentrated. So that's why I put a partition constants higher than 1. And even if we assume that the kinetic the rate constants are the same inside and outside, you will see an overall rate enhancement the more Coacervate phase you have, because of the concentration effect. But what is interesting here is that you see when you go from 0 to 5% volume fraction, you see a rate enhancement. But as you grow too much the Coacervate phase, you get a reversal of the effect. So at very high volume fractions, you lose this concentration effect because you're also removing reactant from the dilute phase. So this is to show that experimentally, in the end, you will only detect the effects of coacervates in a certain region of in a phase space of partition constants and volume fractions. We can do the same for if we assume that the coacervate environment either favors or prevents a reaction. And here, a bit the idea is the same, that you will be able to detect the differences experimentally, but of course you need a high volume fraction to see those. So typically in chemistry, we are looking at the entire emulsion. We collect signal from the whole mixture and not just from the droplets or the dilute phase. And that's why I'm showing here the overall conversion. So, and of course, the effect depends on the concentration of the components in the droplet. What we can do to further complicate a bit things is now include the partitioning of the product as well. I had not taken that into account, but that is important for enzymatic reactions to take that into account, also because there's a lot of product inhibition in enzymatic reactions. So now there's a lot more parameters because the product can exchange with the exterior, and the product can also bind to the enzyme and outcompeting the substrate. So this is the type. So we can make this is a type of product inhibition that is, very common with enzymes. Also very common with self-replication reactions. And if we do the same, so if we assume that that the enzyme is very concentrated in the droplet, and that inhibition is possible, what we can see here is that, so now I'm varying the partitioning of the product. So what you can see is that the partitioning of the product will, taking that into account, will always enhance the reaction rate, but it's like to a point that you reach a situation where there's no inhibition anymore, essentially. And that happens when the product is concentrated outside, so it's excluded of the droplet. So you basically don't have things colocalizing. And on average, product and enzyme are not in the same compartment, and that reverses inhibition. So I find it very interesting to take into account in the reactions that we look at normally. And something else that happens when you take product into account is that you can have gradients. So you can have transient peaks of product accumulation in the droplet, because normally the diffusion is a bit slower. or it's at least comparable to reaction rates. So we can easily find a way to explain gradients in the droplets, for example. But now in the final part, I would just like to move to reactions then in active coacervates. So now that was already complicated, I mean, in my view, at least in passive coacervates, and in active coacervates, it becomes a bit more complicated. So I was interested in this in my PhD because I wanted to mimic exactly droplet growth and division. And this was the way to do it. So you had to come up with a reaction that makes droplet material. And you might think that that's involves a lot of design, but it does not. So what we mean in essentially is we need to just find a reaction that will bring a reactant that has a very narrow phase diagram to a product with a very wide phase diagram. So maybe that sounds very specific, but that's often the case with enzymatic reactions, because to enlarge a binodal, you typically need to increase charge of a molecule, of a polymer, or elongate a polymer. And that's what enzymatic reactions do. They phosphorylate, they polymerize. So I chose the conversion of ADP into ATP. So you add this phosphate here, and that drives phase separation in the presence of a protein. So ADP is soluble with this, completely miscible with this protein, and ATP forms droplets. And this reaction can be catalyzed by pyruvate kinase enzyme using a second substrate here. So I was interested in seeing how the reaction controls the nucleation and the growth of these droplets. So like I just said, the phase diagram perfectly matches what we need. So the phase diagram of ADP is very narrow, and that of ATP is very wide. So you can easily find a situation where the reaction, you start from a one-phase solution, and as the reaction progresses, you enter the binodal of ATP protein mixtures. So when I put that reaction in the microscope, you get something like this. So you immediately start to nucleate droplets, and you see them grow. And if I take the radius of these droplets, it looks like this. So I can make a radius profile over time, and I can follow the droplet count over time. And this type of plot tells us a lot, because it tells us that the number of droplets is increasing. And that's not the case for passive droplets. So passive droplets either stay the same, or the droplet count decreases because of ripening and coalescence. And this also tells us that the radius is increasing in a continuous way, which is also not the case for passive droplets, because they grow by fusion, so very discrete steps of growth. So this tells me that these droplets, they are actually active droplets. And if I look at the growth of all these droplets over time, so they start at very different sizes, but let's just look at them when they have the same size. So if I correct these curves like this, you can clearly see that there are droplets that grow. They grow at the same rate, with the same trend, at least because they are all controlled by the same reaction. And there's a group of droplets that does not grow. And that's because they're so small that they do not have a single enzyme molecule. So these would be the Oparin droplets that are bad at growing, and the other ones are good at growing because they have an enzyme. And this is also the process is completely linked to a reaction because it can be restarted by adding more fuel. So growth, the droplets are no longer active after all substrate is consumed, of course, but you can just restart. So this was, so now I was focusing a lot on the droplet growth, but we can also look at the reaction, because droplet size here is completely connected to the amount of ATP in the droplet. So if instead of looking at radius, I now show you the ATP formation in this system, I get the purple curve here, and I'm showing in comparison what you would expect from Michaelis-Menten kinetics, which is what pyruvate kinase typically follows. And first, well, we get a faster conversion than for Michaelis-Menten, so a faster conversion than expected for a one-phase system. And here I'm also curious to discuss, because at the time we decided to say that this is a type of autocatalysis. So basically that the droplets are, there's this constant, as the droplets, you have a droplet of protein and ATP. So as you keep forming more ATP, it needs to recruit more protein to remain in phase equilibrium. So the reaction drives the growth of the droplet, and then this will also require recruitment of more ADP to establish equilibrium. Again, so this should be essentially an autocatalytic process. Of course, here there is the limitation of the recruitment, of how fast you can recruit molecules. So for ADP, that's not a problem. But for the protein, that's a problem because it's a large molecule. So what we thought is that the purple curve here is perfectly following what you would expect for the growth of any condensed phase. So you have a starting radius, so the volume of the droplet or the amount of ATP. It follows this equation where you have a starting radius and you have a supersaturation rate, and we think this is where the autocatalytic reaction rate appears. But you are always, of course, limited by the diffusion of protein into that droplet. We also, we thought that this is, because it contains this diffusion component, that it would be a type of physical autocatalysis. But of course, now after Alex's talk, I thinking a lot of how we name things. But that's physical autocatalysis is something that is normally shown for lipid aggregates. But I think it just directly applies to coacervates in this case. So just summarize a bit. Among the types of autocatalysis, I think that the most available, or that relates the most to coacervate, to reactions in coacervates, is the physical autocatalysis type. Of course, you can also incorporate networks or template replication inside coacervates. But I think in that case, with the effect you will get in the network will essentially be the one of physical autocatalysis. So I've showed you that some reactions, how rates can change in passive coacervates. There is no autocatalytic effect expected here, but you'll just find in the literature examples like this, where you have a great rate enhancement in coacervates compared to a solution. I showed you my work with the growing droplets, where I think this is a type of physical autocatalysis. There's another work in the literature where they claim that a reaction that is not autocatalytic in nature becomes autocatalytic when Coacervation is allowed. They do not call it physical autocatalysis, they call it concentration-induced autocatalysis. So that's maybe interesting. It looks the same to me. And when it comes to incorporating autocatalytic reactions in coacervates, there's not a lot of work on that, but there's also not a lot of effect observed. So normally you have something like, so a very common example is just putting a ribosome reaction inside the coacervate droplets. And what people are interested is just mainly seeing if ribosome activity is sustained or allowed in the presence of coacervates. And they do not really discuss if the mechanism changes or if the autocatalytic nature is lost. But I think that could be interesting to look at. All in all, it seems that it's just compatible. Also, the ribosomatic activity stays the same in biphasic systems. And the most complex case that I found in the literature is an autocatalytic reaction driving coacervates. So you would expect a very high effect in this case, no? Because it's the most complex one. But interesting enough, so they have an autocatalytic reaction here, and then the product is able to form droplets. And actually, they observe little to no effect by having coacervates or not. So the blue curve is in solution, and the red curve is with coacervates. There's no real effect, and if any, it's slower. The reaction, the network is slowed down by having coacervates, and the greater effect here is in the droplet itself. So it's the opposite effect. So I also find that interesting in the end, that these two cases do not really have show a big effect of biphasic reactions in autocatalysis. So I hope to have showed you that it's difficult to predict the effect of coacervates in reaction rates, and we have to keep doing experimental work to understand this. So quantitative work to get reaction rate constants, I would say. Oparin scenario relies a lot of coacervates facilitating reactions, but we should not take that for granted. It's not like micellar catalysis. It doesn't have to be the case. And we can either incorporate reactions in coacervates. That doesn't seem to have a lot of effect. Or we can make use of the fact that non-autocatalytic reactions become autocatalytic if they are coupled to phase separation. So yeah, it was very challenged by making all these connections, and I'm really curious what you think as well. So that was my part. Thanks.
Thank you very much. That was really really interesting work. So maybe should I stop recording now and we can go on to discussions?